Good morning, everybody. Hello. Hello. Quite clear? So today we discuss the enzymes. So enzymes and properties of enzymes, nomenclature of and classification of enzymes. We discuss in this one. And another topic is the structure and mechanism of the reaction. Is also we discuss that one. Some affecting factors of the enzyme activity here discussed, and enzyme inhibitors is also discussed in this unit 3. So the enzymes are the proteins that catalyze various metabolic reactions of all the living organisms. These enzymes are biological catalysts of the cell occurring in several thousand and carry out the various biochemical reactions which are include the biosynthesis of the protein. So these rate of the synthesis of proteins and biochemical reactions under genetic control only. So the depending on their activity and strongly controlled by the substrate concentration, pH, and temperature is also. They are the exceptional catalysts because they are under the temperature is also. So according to Dixon and Webb, the enzyme is the protein with the catalytic properties due to the power of the specific activation. What are the properties of the enzymes? So the enzymes are needed in the very small concentration to convert the large amount of the substrate to product. Substrate to product. Okay. So here so there are the two terms we are to use. First one is the substrate, another one is the product. These are the designated the starting and ending substances as a chemical reaction. And the respective starting point is the substrate and the product end point is the product. So the enzymes remain unaffected by the reaction which they catalyze, and they are the stable due to protein nature. All these enzymes will spread up a reaction. It will no effect in the equilibrium of the reaction. The specific of the catalyst function of the enzyme is due to its protein nature. The region of the protein which binds with the substrate does the participating in the conversion of the substrate to product. This is known as an active site. Turnover number. So what is the turnover number? Here, the number of substrate molecule converts into one molecule of the enzyme per second. When it is active site, it is saturated with the substrate. The number of the substrate is called catalyzed enzyme. That one we call as enzyme turnover number. The highest turnover number of enzyme is the carbonic anhydrase. Reversibility. Most of the enzymes catalyzed reactions are irreversible reactions are occurred. Okay. The enzymes are very sensitive to heat and temperature. The pH of the requirement is essential. Some enzymes are active in acidic condition, some are the active in alkalic conditions. So some other are the in natural temperatures. So next one is the nomenclature and classification of enzymes. The synthetic, systematic, the nomenclature as well as known is the importance in the study of the enzyme. So most trivial names are given in the nature of the substrate involve the nature of the catalyzed reactions. So most such names with the suffix as ase, for example, dehydrogenase, phosphatase, transferase, synthetase. So according to their reactions, they are catalyzed into different types of the enzymes are there. That is the oxidoreductase. So first one is the oxidoreductase. So these enzyme catalyzed the oxidation and reduction reaction by involving these hydrogen transfer. Okay, so that's why these dehydrogenase, reductase, oxidase. These are the examples of the oxidoreductase enzymes. So second one is the transferases. The transferases are involved in the transfer of chemical group such as the amino group or phosphate group from one molecule to another. The examples is the transminidase, kinase, and phosphorylase enzymes. So these transminase enzymes the transfer amino group from substrate to other molecule. The kinase enzymes the transfer of phosphate from ATP to substrate. And another enzyme is the phosphorylase enzyme, which is the transfer the orthophosphate to substrate. This is the some cleavage bonds and addition of the water is also, that is the phosphatase and peptidase enzyme. So another third the group is the hydrolase enzyme. So hydrolase enzymes means the cleavage bond by the addition of the water. That group we call as the addition of the water molecule. So that is the examples are the phosphatase and peptidase enzyme. Another fourth class is the lyases. These enzymes catalyze the addition group of to double bond or vice versa. Other decarboxylase enzymes, aldolase enzymes, or belongs to the lyases. Another fifth group is the isomerases. Isomerases. Then isomerases means they are involved in the rearrangements of the atoms of the molecule and include the racemases and epimerases, which catalyze the interconversion of the stereoisomerase of amino acids and sugar groups. That one we call as a isomerases. Example is the glyceraldehyde 3-phosphate that is converted to DHAP and glucose converted to fructose. So sixth one is the ligases. So ligases they are linked of the two molecule coupled with the breakdown of ATP. The energy is liberated by ATP is used for these condensation. These enzymes are involved in some important reaction like linkage of amino acids to RNA. That is the classification. So The next one is the enzyme structures and mechanism of the enzyme. You see here in this one, the enzyme structure. So the molecule weight of the enzymes around the 10,000 to 1 million and contain many thousands of amino acids. The substrate molecules are very small compared to the enzymes. So only the small part of the enzyme will be the contact with the substrate enzyme substrate complex. So the catalytic ability of the enzyme is dependent on the three-dimensional shape. The catalysis of an enzyme catalyzes the reaction is consists of three steps are involved in this. So the first one is the substrate recognize the enzyme and to come together on the specific arrangement. That one we call as the enzyme substrate complex. By the catalytic activity of the certain group of the enzyme surface, the substrate will be converted to product. The products leaves the enzyme. This is so to explain the mechanism of the action of the enzymes here, two theories are there. So based on their activity, based on their reaction, then. So first one is the lock and key theory. Lock and key theory. So this theory is proposed by the Emil Fischer. Emil Fischer, first of all, this is proposed by the lock and key theory. As only the specific keys fit in the particular lock to open. Similarly, a specific substrate combines with the active site of the specific enzyme to form the enzyme substrate complex. The enzyme substrate complex breaks after the substrate has been converted into product, and the enzyme again it will be free. So that is the lock and key theory. Means what happened here? A specific structure is used to a specific enzyme. The mechanism of these chymotrypsin action shows the chymotrypsin is the digestive an enzyme which catalyzes the hydrolysis of the protein in the intestine of the human beings also. This is the lock and key theory. So next one is the another example is there, that is the coastland induced fit theory. Coastland induced fit theory. So this theory is proposed by the Coastland. That's why the name is given by his name only. Coastland induced fit theory. According to this, the active sites are not active. Only when the substrate is approaches the active site of the enzyme, it is induced some conformational changes, configurations, changes in the enzyme, that is the facilitating the reaction. So according to this Coastland theory, so the active sites are inactive condition when the substrate is come together and enzyme, that active sites are in the active. As the fit it is induced by the substrate is called induced theory. Okay. So next one is the factors are affecting on the enzyme activities. So what are the factors already discussed that one? The substrate concentration, enzyme concentration, temperature, pH is all are the affecting the enzyme activity. First of all, we discuss the substrate concentration. The enzyme catalyzes the reaction source and the saturated affecting with the increase the substrate concentration when the enzyme concentration is kept to constant. At a very low concentration of the substrate, the rate of the reaction is very low. But in these reactions, to increase the substrate concentration, the reaction will be increased. So further increase the substrate concentration will cause only a marginal increase in the reaction rate. So finally, a point will be reached behind which is the reaction rate will be approached, but never reach the plateau, the maximum rate of the reaction. The next one is the enzyme concentration. Okay. Enzyme concentration here, it is affecting on the different concentration of the enzyme and reaction rates of the enzyme activity. The low concentration of the enzyme is used with the high concentration of the substrate. The rate of the reaction is directly proportional to the enzyme concentration. This one. Another one is the temperature. For the majority of the enzymes within the temperature up to 37 degrees only, usually the rate of the chemical reaction is approximately double. For every 10 degrees centigrade, it will increase. This is called Q10 of the reaction. So what happened here? Q10 means every 10 degrees temperature will be increased, the reaction will be doubled. So with this regard, the enzyme is catalyzed reaction. This will be followed up to 25 degrees. The increase the reaction rate due to the increase the kinetic energy, both substrate and enzyme molecules, and this will be result increase the changes of the collection. Okay. Between the enzyme and substrate molecule, the majority of the enzymes have high activity between the 25 to 37 degrees. So another one is the affecting the pH. What is the pH here? The enzymes are very sensitive to small changes of the pH. Many enzymes have the maximum activity at the near to 7 pH. Their catalytic activity decreases at the pH is shifted away from these optimum. So the pH curves is the different curves are there. The pH with the extreme values bring about the changes in protein substrate that later the stability of the enzyme. The pH changes is the ionization of the enzyme, thus affecting the affinity of the enzyme for the substrate. It alters the ionization of the substrate and affects the binding of the substrate to the enzyme. The pH changes is the ionization of the enzyme substrate complex. This is the so another one is the inhibitors of the enzymes. The enzymes. The compounds which have the ability to combine with the certain enzymes and block the catalytic protein, that one we known as the inhibitors of the enzyme. So there are the two types of the inhibitors are there depending on their reactions. That is the irreversible inhibitors and reversible inhibitors. So what are the irreversible inhibitors? Means the irreversible inhibitors from the covalent bond with the specific function group, usually an amino acid residues, which is the association with the catalytic activity of the enzyme. There are some inhibitors which are the physical block of the active sites. So that is these inhibitors cannot be removed by the dialysis with the usually means that the functional group of the enzymes molecule have been destroyed. So this is the another one is the reversible inhibitor. So reversible inhibitors means the enzyme active can be restored by the removing the inhibitor by the dialysis. So these inhibitors involve in the equilibrium between the inhibitor of the free enzymes, which can only be explained by the that is the Michaelis Menten kinetics. That is. So these reversible inhibitors, they are classified again into two types are there. There is the competitive inhibitors, another one is the non-competitive inhibitors are there. So competitive inhibitors means the compounds that compete with the substrate for the binding to the active site, but once bound and cannot be transferred by the enzymes known as the competitive inhibitor. So another one is the non-competitive inhibitors means the compounds that is the reversible bind with the either enzyme or enzyme substrate complex known as the non-competitive inhibitors are there. Okay. So in the example, this inhibition of the L-tyrosine dehydrogenase by the L-isoleucine and cyanide. So this is the enzymes and classification and nomenclature. Another one is the enzyme structure and mechanism of the enzymes. And another one is the some affecting factors of the enzyme activity. This one. So next one, we discuss that one, the photosynthesis. Photosynthesis means this is the second block. Okay. So photosynthesis here. So photosynthesis. So what is the photosynthesis means? So we know that one. Almost all the living organisms on this planet, depending on the directly or indirectly on the solar radiation, we know that one. Okay, depending on the solar radiation, they are converted to and different forms. They are conversion of the radiant energy into chemical energy by the plant, known as the photosynthesis. What is here? The solar energy, solar radiant energy converted into chemical energy by the process of photosynthesis by plants only. Okay. So the overall process is the oxidation of the water to produce oxygen and reduction of the carbon dioxide to carbohydrate. So here two processes are occurred. They are undertaking means oxidation and reduction. Oxidation means the water is oxidized to produce oxygen and the carbon dioxide to reduce that is to form the reduction and to form the carbohydrates. Okay. So the carbohydrates are turned into the liquids and molecular nucleic acid, proteins, and other molecules to form the several uh, another biochemical process are there. So here in the reaction here 6 carbon molecule plus 12 H2O water molecule in the presence of light and chlorophyll to form the glucose plus 6 H2O. And this is the reaction of the photosynthesis, where the C6H12O6 is represent the simple sugars, that is the glucose or sugars. This is the light driven and the redox process are there. So next one is the structure of the chloroplast. So structure of the chloroplast means so that is the photosynthesis takes place in the discrete, the highly organized subcellular organelles that known as the chloroplast. The chloroplasts are generally ellipsoidal with the diameter of the so the chloroplasts are generally ellipsoidal with the diameter around the 5 to 10 millimicrons. They are mainly found in the mesophyll cells only. Okay. So the structure of the chloroplast have been studied in several scientists where they are discussed. So that's why they are undertaking the chloroplast is there. So that's why the plants are called as autotroph. Okay. So here there are the four important structural components in the chloroplast are there. Each chloroplast is surrounded by the double membrane known as envelope. Within the chloroplast, amorphous get like the enzymes, rich material is present. There is called as the stroma. Okay, stroma, that is we call it, and which contain the enzymes which are involved in the reduction of the carbon dioxide and other enzymes and metabolic of other biochemical pathway, that is the DNA and RNA synthesis pathway. The pigments contain the thylakoids are embedded through the stroma. In some areas, their adjacent thylakoids appear to closely appears. They give the rise to stalk called grana. The thylakoids with the grana, known as grana thylakoids. Some thylakoids they are extend behind the grana stalks into the stoma as a single non appreciated membranous structure called as the stroma thylakoids. Then the thylakoids contain chlorophylls and carotenoids are there. The carotenoids, some sites are extended to the photochemical reaction. The activity between these two membranes, they are thylakoid, that is called as a lumen. Okay, and this is the site of the photosynthesis of water and the source of the proton involved in the ATP synthesis. So next one is the chlorophyll pigments. So here these pigments which are associated with the photosynthesis, they are classified into three groups are there, that is the chlorophyll, carotenoids and phycobilins. So here according to the chlorophylls, they are in different types of the chlorophylls are there. So chlorophyll A, that is the universal pigment. Chlorophyll A we call as the universal pigment, and which is present in all autotrophs except photosynthetic bacteria. All autotrophs means all the photosynthetic plants conducting the photosynthetic plants except photosynthetic bacteria. So next one is the chlorophyll B, which is present in the chlorophyll and all the higher plants. Then chlorophyll C, this is the phaeophyce and bacillariophyce. And another type is the chlorophyll D, which is present in the rhodophyce class only. Means these are the some algal members: chlorophyce and phaeophyce, bacillariophyce, rhodophyce. And chlorophyll E, which is present in the xanthophyce class, example Vacheria. And chlorophyll A and chlorophyll B, these are the present in the photosynthetic bacteria. So here some green bacteria also contain the chlorobium 650 and chlorobium 660. Okay. So another one is the carotenoids. So carotenoids means the thylakoids also contain the certain carotenoids apart from the chlorophylls. The color ranges of these pigments is varies from yellow to orange. As these pigments are found in the large quantities in roots of the carrot, they are designated as carotenoids. The carotenoids contain an open chain is conjugated double bond system ending with the iodine rings are there. So these beta carotene are present in the carrot root, and lycopene carotene is present in the tomato. This is the specific carotenes are there. So these carotenes serve as a dual function, that is collecting the radiant energy that can be used in the photosynthesis and protecting the chlorophyll from the photoradiation. So these carotenoids are generally we call as a accessory pigments. Accessory pigments means additional pigments we call it. Another one is the phycobilins. Another type of the pigments here, phycobilins. These are the water soluble pigments present in the Cyanophyce and red algae. Red algae members means rhodophyce and Cyanophyce. Chemically, these resemble to the chlorophylls to avoid the magnesium with the four pyrrole rings. The stated magnesium ring is absent. So phycobilins is resemble the bile pigments, which are the degradative compounds of the porphyrins. There are four types of the involved in the photosynthesis known as the phycocyanin, phycoerythrin, and allophycocyanin. Another one is the a special type of the pigment is the phytochrome. So it plays an important role in the several photomorphologic process. Phytochrome. This phycobilin pigment is differ from the chlorophyll in that the tetrapyrrole group covalently linked with the protein that form a part of the molecule. This is the pigments. And answer one is the light absorption spectrum. So all the energy from the sun to reach the earth to arrive in solar radiant, a part of the large collection of the energy then called as the electromagnetic radiation spectrum. So this solar radiation include the radio waves and the infrared visible light and ultraviolet and X-rays and gamma rays. So they are the visible light and wavelength, that is the VIBGR. We call as the VIBGR means violet, blue, green, yellow, orange, and red. That one we call as the VIBGR. So wavelength of these violet here 380 to 450 nanometers, and blue so here that is the chlorophyll A. The chlorophyll A is the maximum absorption of the blue light, that is the 435 nanometer, and the maximum absorption of the red light here 673. So this is the, then chlorophyll B, the maximum absorption of the blue light here 435 nanometer, and maximum absorption of the red light here 642 nanometers. So carotenoids they are observed the 449 nanometers and 478 blue light and they are to observe. So another one is the mechanisms of the reaction. Okay. So here the mechanism of the photosynthesis. There are the two stages are there in the photosynthesis. That is the light reaction and the carbon assimilation stage. So the light reaction, the sunlight is energy converted into NADP and ATP. That is the chemical energies are there. This energy is called as assimilatory power. This reaction takes place in the thylakoid membrane of the chloroplast. So another stage is the carbon assimilation stage. That is the light reaction. That is end product of the NADPH and ATP is utilized in the carbon assimilation stage in order to exchange the carbon dioxide into carbohydrates. This reaction takes place in the stroma of the thylakoid, means chloroplast. Okay. So here there are the some reactions are there. That is the Hill reaction and Emerson enhancement and Z schemes are there in the photosynthesis. What is the Hill reaction here? The Hill reaction means which is developed or to identify the robot hills only. That's why known as a Hill reaction is called. So Robert Hill is demonstrated that the isolated chloroplast when the elimination in the presence of artificial electron acceptors such as the ferrocyanide release the oxygen and this phenomenon is referred as a Hill reaction. So what happened here? Isolated chloroplast which is eliminated the artificial acceptor, electron acceptor. With this experiment, hills provide the water is the source of evolved oxygen. So that is the another scientist is Rubin and Kamen in 1941 using the oxygen 18 isotope had shown that is the source of the oxygen is evolved during the photosynthesis from the water. Based on this experiment, the photosynthesis is regarded as a photochemical reduction of the oxygen here. So another one is the Emerson enhancement effect. Robert Emerson and his research group is working about these enhancement effect in the chlorella. So what happened here? They are the found the wavelength longer than the 680 was applied, the photosynthetic rate will decrease. What happened here is applied the 680 nanometers, so that is the photosynthetic rate will be decreased. That stage we call as the red drop. When the immersion is applied only 653 to 680 nanometer photosynthetic, the rate will be increased. They observed that the light of these start wavelength around the 650 and the longer wavelength 680 were used in simultaneously. The photosynthetic rate was higher than the sum of the two rates with the separate beams. This enhancement we call as a Emerson enhancement effect. So based on these observations, the Emerson suggests that the photosynthetic involves the two photochemical reactions, one driven by the wavelength here 680 and another short wavelength that is the 650. They are designated as photosystem 1 and photosystem 2, depending on the wavelength, only. So here there are the two photosystems are physically and chemically distinct in each the own antenna, pigments, and photochemical reaction centers. These two photosystems are connected by the electron transport chain. That is the Phil and Benland is proposed the model of for the photosynthetic electron transport. This is the scheme. That one we call as a scheme is called as a Z scheme. Photosystem 1 and photosystem 2, they are involved in this uh, system. So in Z scheme, there are the two centers present. They are the photosystem 2 and photosystem 1. When the thylakoid membrane are treated with the mild detergent like sodium docatyl sulfate, and after the ultracentrifugation, it is observed in the complex thylakoids. So that is the cytochrome photosystem 2 and cytochrome B6 and F complex, photosystem 1 and ATP synthesis. The photosystem 2 consists of the core complex of the six integral polypeptide and the reaction center, that is the 680. These polypeptides are coated by the chloroplast genome. The most important polypeptides are the D1 and D2 with molecular weight around the 32 and 34 kilodalton. The photosystem 2 complex also contains three peripheral polypeptides coded by the nuclear genome. These polypeptides are involved in the binding with the calcium ions and chlorine. Both are essential for the photolysis of the water. Okay. So in these photosystem, 680 nanometers is receive light energy, which is inductive resonance about the 250 chlorophyll A and chlorophyll A molecule, and numerous carotenoids also involved. Okay. So this is the Z scheme, means photosystem 1 and photosystem 2. Yeah. So cytochrome B6 and F complex. What are the B6 and F complex? Here mean this one is the plastoquinone diffuses laterally after this is released from the reaction center of the photosystem 2 until it is encountered the cytochrome B6 and F complex. This is the multiprotein membrane spanning complex, though this principal constitutes the cytochrome B6 and cytochrome F. It also contains the non-heme iron protein means heme iron protein, only the risco iron sulfate protein. Okay. So next one is the photosystem 1. So here the photosystem 1, they are observed the light energy independently of the photosystem 2 and contain the separate core complex are there. So the main function of these photosystem 1 is transfer electron from the plastocyanin to ferridoxine. The photosystem 1 complex contain the number of bound electron carrier, and the most extensively characterized one is the uh, 700 nanometer P700. This pigment undergoes light-induced oxidation to produce the P700, and the electron lost from the P700 is transferred to the ferridoxine. A low molecular weight is iron sulfur protein, and the electrons from the ferridoxine are finally passed to the NADP to form the NADPH. This reaction is catalyzed by the enzymes by the ferridoxine NADPH oxidoreductase, and the photophosphorylation, that is the That is the ATP synthesis is called as the ATP synthase or photophosphorylation. So, photophosphorylation means here uh, that is the another important complex in the thylakoid membrane is ATP synthesis, which is convert to ADP and inorganic phosphate to ATP. Okay, it is the exchange along with the photosystem one stroma thylakoid. ADP and inorganic phosphate then convert into ATP in the presence of ATP synthase, which is located in the thylakoid membrane only. Okay, so the ATP synthase contains the two parts, that is a stalk known as the CF0, and that is extended to the lumina to the stroma across the thylakoid membrane, and spherical part is called as the CF1. Means head and tail is there in the ATP synthase, like that. See here. Uh, okay. So the molecular weight of the ATP synthase is around 400 kilodalton and is known as a coupling factor. The photophosphorylation was discovered by the Daniel Arnon in 1954. The ATP synthase caused the ATP formation in the stroma, which is associated with the transfer of hydrogen from hydrogen ions, that is the thylakoid lumina to stroma. These protons, nothing but protons in the thylakoid lumina, arise from the oxidation of the water and is PQ2H2 means hydroquinone. This oxidation results in the hydrogen ions means the protons concentration to lumina around the 1000 times greater than the stroma. So the concentration will be increased. So although thylakoids are impermeable to hydrogen and other ions, the ATP synthase is involved in the transport of the hydrogen ions from lumina to stroma through ATP synthase. So this is the Mitchell in 1961 proposed the pH gradient could be provide energy of the ATP formation. That is, his hypothesis was commonly known as the chemoosmotic theory, which is proposed by the Mitchell. So chemoosmotic theory that is suggested the energy of the ATP synthesis is derived from the proton motive forces. The proton motive force is some of the proton gradient and transferable electric potential. These is hydrogen, these protons to ATP still the recently was considered to the there are the protons are there. Four protons means three protons and four protons are transferred to across the membrane for the synthesis of one molecule. Therefore, the ratio of the hydrogen atoms and ATP's gradient as 4 instead of the earlier 3. So in previously These three protons are used in one ATP. But nowadays, the revised recent researches, four protons are used to synthesis of one ATP. So there are the 8 protons are required to oxidize to water molecules. 8 protons are continuously supplied from the water and withdrawn the NADPH. This formation of the ATP is in the non-cyclic electron transport known as the non-cyclic photophosphorylation. Is photosystem 1 may be also transport the electrons back to plastoquinone plastoquinones rather than the NADP. This formation of the ATP by this electron transport pathway is known as the cyclic photophosphorylation. Okay, so this is the photophosphorylation, the remaining ATP synthesis, and some pigments we discussed in this one. Okay, but we discussed that one in photosynthesis, the chloroplast structure. Discuss that one. Some types of the pigments means chlorophyll, carotenoids, and phycobilins, and phytochromes. Also, we discussed that one. And light absorption spectrum we discussed. Visgia we discussed that one, and some mechanisms are there. And Hill reaction, immersion enhancement, and jet scheme we discussed in this photosynthesis. Okay. Okay. Thank you, everybody. And another class we discuss tomorrow. Okay. Thank you.